So Anna, who's over there with the camera, and Maurits, who is over there at the front, and myself, Joshan, we've organized the Psychedelic Science Collective to start giving lectures on psychedelics. We're now also organizing journal clubs in psychedelics and some social events every now and again. So at the end, I'll tell you what's coming up later. But for now, we're very happy to introduce to you the second speaker of this academic year, which is Fejer. And we're very happy to have him. He is one of the most active members, I think, of APRA, which is the Amsterdam Psychedelic Research Association, which is basically doing similar stuff in Amsterdam as we are doing in Rotterdam. So they just have sort of a 2-year head start to what we are doing, so they progressed quite far. And George will show like all the intelligent ideas that they came up with already. So I think it's going to be a very interesting lecture. I will not explain the title, I think, because probably George is going to do it himself. So I'll just end with, it's called The Doors of Prediction, Psychedelics and Predictive Processing. Have fun, and I think one thing, because it's going to be an hour, right? About an hour.
Is time an issue?
No, it's not an issue, but like, do we need a break or not?
Maybe. It will be indicated where we can take a break.
Okay,
like,
we'll see how George's pace is and like how awake you are, yeah?
Yeah, let's do that.
All right, great.
Okay, hi everyone, thanks for the nice introduction. Like Josiane already said, I came all the way from Amsterdam to give you this lecture, and this is basically, just based off the work that I did for my master's thesis, which was one of the very first placebo-controlled studies on the effects of microdosing that was conducted at the University of Amsterdam. But there is also a bit of a theoretical background that I want to just walk us through and give you all a sort of basic understanding of where I'm coming from and where the theories that I work with, what they are, and what is predictive processing, and what do they have to do with psychedelics. So let's just jump right into it. So what is predictive processing and how does this relate to the psychedelic state? Well, I always like to give a brief introduction about what we actually know about psychedelics and what they do to the brain. So this is one of the older studies from 2003 where they looked at the different EEG bands, and they found before versus after psilocybin infusion that it just massively desynchronizes all these different networks that are that you can see in, based on the different brain rhythms of the EEG state. So you have alpha rhythms and beta rhythms, delta, low gamma. Now these are all the different frequencies at which the brain sort of communicates, the basic idea is that synchrony is the brain's currency for transmitting information, and that these synchronous activity patterns are sort of transmitted at different speeds and different frequencies. And all of these networks become desynchronized under the influence of psychedelics. So you see a decrease in the oscillatory power. And when you look at the different fMRI studies, which measures the Bolt signals, so the blood flow to the brain, you basically see the same things, that not only do the cortical layers of the brain that you can measure with EEG get desynchronized, but also like the underlying neural networks that you measure with EEG get desynchronized. Like for instance, the default mode network, which has been correlated to ego dissolution. Now this is one of the main theories of the entropic brain theory, which I will be talking about in a moment. But what they basically did was that they saw that the default mode network becomes very unstable, very random and chaotic, and the degree of this randomness was sort of correlated with the phenomenology of ego dissolution. So that is why they think, it inhibits the ego. And by now this must be one of the more famous or most aesthetic depictions of what is going on. If you want to tell somebody what psychedelics do in one single nice graph. And you see what they basically did was they took all the different network hubs and they plotted their overall average connection to one another. So what you see is that when you give people psilocybin compared to the placebo condition, you have all these different brain regions that are communicating with each other. That is usually not happening. But how do we make sense of this and what is happening? Well, like I said, there's a decrease in the intrinsic connectivity within different networks, but you have an increased connectivity between different neural networks. So, those are just like the state-of-the-art findings of what we know about psychedelics currently based on the neuroimaging literature. Now I want to go talk about, first of all, I want to explain what the entropic brain theory is. And basically, this idea that the brain is disorganized can be described mathematically through the analogy to entropy, which comes from thermodynamics. Entropy describes the orderliness of a state when you describe like a collection of gas molecules. The more entropy you have, the more heat, the faster the particles move, and the more chaos and disorder that introduces. And this is just kind of a mathematical way of describing a physical system, which of course, the brain is also a physical system. So the idea so this idea basically just states that, different states of consciousness can be described across this continuum where you have certain states of mind, like the psychedelic state, or psychosis, or magical thinking, or divergent thinking, that can be attributed to high levels of entropy, so high levels of chaos and disorder, whereas you have other states that are described by almost too much order, so like a very rigid state, think of like a coma, or sedation, or deep sleep, or depression, or psychopathologies that are sort of marked by repetitive behaviors where people get stuck, and it's like a very simple idea that basically their brains have these very predictable and rigid set of states that they are sort of stuck in, right? So moving on. What's not happening? So what kind of theory could we use to describe what is actually happening? This is where predictive processing comes in, but before I go there, I want to take a step back and maybe approach this problem from a bit of a more philosophical perspective. Like, why do we even need unified theories? Like Freud was the last person who put out something that he tried to call a unified theory, and now we have the entropic brain theory and words like ego dissolution that are coming back. It's kind of, in modern science, it's kind of frowned upon to bring Freud out of the closet, and this sort of translates to people not really wanting to indulge in grand unified narratives anymore. So I'd like to take a moment to ask this question: why do we need theories in the first place? You could also argue from a sort of data-driven neuroscience that we do not need any theories, we just need to get more and more data, and that we are primarily limited by the amount of data that we get. We just need larger and more complex multimodal data sets to understand the brain. And as soon as we found enough ways to measure it, we're just going to understand what it's doing and what psychedelics are doing. But I'd like to challenge this notion by saying that such data sets, first of all, do not exist yet. Eventually they might. But if they could we actually evaluate how these complex systems worked? Now, this was the question that was asked in this very elegant paper from computational biology, where the scientists looked at whether a neuroscientist could actually understand how a microprocessor works. Now, unlike the brain, we know that a microprocessor has a very distinct set of functions. We know that it has a purpose because we programmed it, we created it, and it can simulate a lot of complex behaviors. Here they took a Commodore 68, an old school computational unit, and they looked at the simulation of complex behaviors, namely you know, these old arcade computer games like Donkey Kong Space Invaders, Pitfall, Asteroids. So basically an engineered model organism. And they wanted to find out whether a neuroscientist with all our fancy modern neuroimaging methods could you know, evaluate what this organism was actually doing. And you know, they went through the entire neuroscientific closet of techniques. So they did a microprocessor connectome project where they put the microprocessing unit under a microscope and reconstructed the whole thing digitally. So they created like a virtual model of the unit. Then they looked at the connectivity and cell types and characterized various parts of the microprocessor as like a neurobiologist would do to look at what kind of different cells are doing what kind of different behaviors. They looked at transistor lesions. This is what you often would do with a TMS coil, where you inhibit certain parts of the transistor and see how that affects the behavior. They looked at spike tuning. So again, they looked at individual transistors and tried to see how its activity correlates to the behavior that is being measured. They looked at local field potentials. This is going towards the kind of techniques employed by EEG. They looked at activity mapping. This corresponds to basically what you do with the fMRI. And they looked at sort of these causal filter models. So this is where you take all the data and, you know, put it through a mathematical filter that tries to tell you how much of that data is actually causally linked to one another. And, you know, in the end, this is what a neuroscientist does. You come up with these very highly complex sort of causal maps of what the brain is doing. But, you know, this doesn't really correspond to an actual explanation of what is going on. And, you know, there's not much attention being paid to the phenomenology of what you're trying to explain. So, you know, in a nutshell, no, a neuroscientist could not explain what a microprocessor is doing. It is clearly not just a limitation of having enough data, because here they have access to all the data, potentially. But, you know, on a fundamental level, the neuroscientist could not explain what the microprocessor was doing. So in a way, these approaches reveal interesting structures in the data, but do not meaningfully describe the hierarchy of information processing in the microprocessor. And so you know, the current analytic approaches in neuroscience may fall short of producing a meaningful understanding of neural systems, regardless of the amount of data that you get out of it. Right? So this is where, in my opinion, predictive processing comes in. It sort of serves the function of having a sort of meta-narrative or of something that the brain is fundamentally doing, sort of the equivalent of an evolutionary theory. But it serves the purpose of not just, you know, analyzing the data, but actually approaching it in a sort of theoretical manner. So let's go on. What are these theories? Let's start off with the Bayesian brain, and I'm going to draw some links to the Doors of Perception by Aldous Huxley. In doing so, now Huxley wrote in his book that there are the things known and there are the things unknown, and in between are the doors of perception. Now, already here in this passage, you see that Huxley is referencing this idea that there are certain structures imposed by the brain, which is, you know, our prior knowledge, and that there are things that are the unknowns, like those are the things that the brain is trying to sort of predict based on the things that it's already learned. It's sort of imposing these structures on the world and how you view reality through the things that it already knows. And this is the idea of the Bayesian brain, that, you know, we have this influx of incoming sensory information, and we're just using our prior knowledge to constantly predict future outcomes. Now, in his, as Huxley goes on in his book, which describes his journey while taking mescaline, he describes these sort of mystical moments, particularly when he's looking at the red-hot poker flower. And he writes, "A moment later, a clump of red-hot pokers in full bloom had exploded into my field of vision, so passionately alive that they seemed to be standing on the very brink of utterance. The flowers strained upwards into the blue." So, as you see, he took this psychedelic and suddenly, all these features of the flower appear to him like much more novel and much more infinite than they did before. And you can draw an analogy to natural hallucinations that people undergo while they're going through sensory deprivation. So when you put people in these sensory deprivation tanks, you also see that they are deprived suddenly of sensory input, and suddenly the brain starts hallucinating mental imagery. And how can this be explained? Well, some of the very earliest observations on sensory deprivation tanks gathered this sort of first-person data from the participants. They say our subjects' hallucinations usually began with simple forms, then visions became more complex with abstract patterns, repeated like a design on wallpaper, or recognizable figures such as rows of little yellow men with black caps on their mouths open. So as we see here, these hallucinations aren't completely random, but they usually start off from some kind of feature space with like simple forms. Then they gradually become more and more complex, and then at the end you see like very complex things that are happening. So this already alludes to the notion that there might be some kind of structure within these hallucinations. Not just random things being hallucinated. But maybe this can tell us something about the feature space in which they occur. So you can go from a simple feature space to a more complex feature space. And this already gives us some kind of notion of what predictive processing is trying to explain on a computational level, namely, what is the problem at hand? Now, how many of you have seen this image before? So maybe you can tell me what is on the image then? Yeah, it's like a Dalmatian. So maybe if, yeah. So I can't reach over there. So maybe there's a pointer. Yeah, so here you can see that this is like the ear, and this is the snout, and the here's one of the legs. It's one of those classical Mooney images where that the prior knowledge affects how you perceive it. So this tells us something fundamental about the brain, insofar that once you know that it is a dog, you can never unsee it, right? So having that prior knowledge that this is actually a dog, it means you can never view this picture in the same way. And what is basically telling us that the problem at hand that we are trying to explain with our theory is the problem of handling uncertainty. And what this means is that the brain is just fundamentally trying to extract some property of a noisy stimulus that tells us what actually caused it, right? And and then the interesting thing about that is when you take a model of something, of a complex phenomena that is for example, in this example, natural speech, what you can do is that you can extract so this is what like regular, so this is what normal speech looks like. It's a very noisy pattern, and through some complex filtering, you can extract the so-called speech envelope, where you take like the most basic characteristics of the speech and just filter out all the noise. Now you'd think that having gotten rid of all that complexity, this should be very easy to understand. But when we listen to the speech patterns, how many of you understood what that was? So probably no one. Now something interesting is going to happen. I'm going to show you what the more complex form of that speech pattern sounds like.
It was a sunny day and the children were going to the park.
Okay, now let's listen to the other one. Yeah, so now everybody understood it magically. Now what just happened in between, right? Like I gave you the prior knowledge of of this thing, and you instantaneously learn to perceive it differently. So, I just changed your consciousness right in that moment. Pretty cool. Okay, one more.
It was a sunny day.
Okay, on we go. Yeah, so so moving on, we now know what the problem is, but then how do we solve it? And this is, sort of where, Bayes' theorem comes in. And, and Bayesian statistics are a nice way of actually formalizing this problem in a much more rigorous mathematical formula. And it can be clearly illustrated by a simple example of what Bayes' theorem is doing. Namely, when I go outside, when I went outside today, I wanted to know, looking at the gray clouds, if it's going to rain or not, if I need to take my umbrella with me. But this is not like this is like a simple statistical problem where I can evaluate the incoming information to make a rational judgment of what I should do. But this becomes a sort of different question if I take the environment into account as well. So what Bayes' theorem is doing is that it takes in the conditional probability into solving that equation. So it doesn't only depend on whether I see the gray clouds, but it also depends on where I'm seeing those gray clouds. So, maybe it might rain less in Amsterdam than in Rotterdam. And nice thing is that this is exactly what Bayes' theorem does. It gives me that exact estimate that I'm looking for. How likely is it that it will rain given that there are gray clouds in Amsterdam or in Rotterdam, right? And I can just implement these different variables into my theorem. So now we have, so now we are 2 steps closer to describing what the theory is doing. Namely, on the computational level, we're trying to describe it's to reduce uncertainty based on the prior knowledge, and we have a nice mathematical formula for doing so. So, like I said before, our perception or our decisions basically depend on the sensory evidence, and we can just implement as many priors as we want. So we can just take all these different conditions into and implement them in this mathematical formula. But there's one problem left, namely the implementational level. And how is this achieved? Well, the brain cannot possibly compute and integrate all these variables simultaneously. So even though I have a nice mathematical way of creating a statistic around how likely it is that I'm going to see something at any given moment based on these and these environmental variables, there's no possible way that the brain can function as one huge processing unit that just simply takes in all these priors and mashes them together in one go. It needs to have some kind of realistic way of implementing it biologically. And this is where predictive coding comes in, and this is why many people are calling this sort of the next unified theory, because it actually gives a very concrete idea as to how these are implemented within the brain, realistically. And it says that the brain just has 2 different streams of information. There are like there's the sensory stream of the things unknown, and there's a second stream, the backward stream, that is based off prior expectations. And what happens is that as the information comes in through our senses, it creates different levels at which the prediction error is computed. So it makes a prediction on the first level, and whatever is not explained away is just sent on to the second level, and there it does the same thing. So it sort of partitions it and creates like different temporal gradients where the information is sort of predicted in this kind of Bayesian manner. So you don't have to like implement all your variables into one long equation, but rather there's like different brain hierarchies that are temporally structured. And this is like actually a very efficient way of compressing information, because the brain doesn't actually have to pass in all that sensory information. The only thing it is actually paying attention to is what it cannot predict. So all that brain activity, according to this This theorem would postulate that the brain is only paying attention to the prediction errors, right? It also says that it has these predictions have a nice hierarchical organization. The expectations are distributed across separate levels, and predictions from higher areas explain away sensory input. And overall, the goal of this processing system is to minimize the prediction error. So, you know, our brains are constantly trying to infer hidden reality states. But what are, so of these so-called hidden states, and what this basically means is that the brain is not sort of directly perceiving reality, but rather trying to make a perceptual inference constantly of, you know, what is it that caused me to perceive this and that. You know, and these are kind of very much embedded into the environment, so it does mean that the information that we receive from our environment plays like a pivotal role in this, and we can basically see that based on these different you know, perceptual illusions. So when we take a look at these fruit baskets and turn them around, we suddenly see faces, right? This is because we have like a very strong sort of prior that always expects us to see faces, which is why, you know, we can create these wonderful works of art where the fruit bowl, and even though you didn't see the face when it was turned around, right? Because you always see faces in one direction. So this means that your prior is sort of trained to perceive the world in one certain way, even though the information is exactly the same. You can also create these other nice perceptual illusions, you know, where you know, space this is to show you the role of space in all of this. So this looks like a guy sitting next to standing next to the lake, but what is he actually doing? Yeah, it's actually a tree stump. And also one of my favorites, again, you see two faces. They appear to be identical until you turn them around, and they're different. So, you know, this is just to show you, even though you know, it's the same exact information, there was nothing different except the orientation, but, you know, your perception of these objects radically changes when they're not, you know, in the way that you process it naturalistically. And when you put and what this means is that you have like these sort of perceptual priors, like the faces or like the orientation of certain objects, and they are represented as your sort of expectations that try to minimize the prediction error, right? I think we went through that pretty well. What is also nice about this model is that you can, as I said before, you have these nice way of, it makes it very simple to model what is going on, because you have a sort of Gaussian distribution about the likelihood of your prior expectation, and then you have the incoming sensory information, which is then again represented as a second Gaussian, and then you have the outcome of that process, your perception, is basically what comes out in the middle. And the prediction error then sort of just goes back and to update these prior expectations. What is also good about this is that you can sort of capture the role of attention in this model based on the role of how precise or how certain you are of those expectations. And this is just to show that there's like always a balance between the prior knowledge and the incoming sensory information, which is captured, you know, in the way that the Gaussian is distributed. So when you have a sort of high precision sensory information, then your perception is more drawn towards the incoming information, whereas when you have you're very certain about your expectations, you know, that could also even force you to blindly categorize the incoming information in a manner that fits your prior expectations more than it should. Yeah, but when you plug in psychedelics into this thing, it basically comes down to the level of detail. And now what this means is that when you have a certain expectation, there's also an appropriate amount of detail that goes along with these sort of predictions. So when you go into a forest, you might predict like certain trees or like plants or animals. But then when you give somebody a psychedelic, the idea behind this theory is that psychedelics increase the level of detail drastically. So you no longer just expect trees or animals, but you expect like certain types of trees or certain types of animals. Your expectations become much more detailed and distributed, thereby increasing, the amount of prediction error drastically, because you're making far more expectations in this state. And as I said before, this is because this is like this interplay between internal and external entropy. You have like a far greater likelihood of misclassifying certain incoming signals. So when you're going in the forest and tripping on some mushrooms and you're looking at that tree stump, it might suddenly turn into an elf. And that is because you have these sort of much more detailed priors that are making predictions that wouldn't occur regularly. So the noisy data then becomes misinterpreted with like with one of the priors. And finally, I come to the free energy principle, which is like the latest edition of this theory. And what this basically says is that you can also minimize this uncertainty through action as well as perception, because before before perception only played a role in terms of a passive process where you just receive the incoming information and you update your priors. But what the free energy principle says is that you can also basically test your environment. So if you're not certain about something, you just sort of test different hypotheses by playing around with the object or sampling the information differently. So this actually means that you can participate in this process actively in reducing the uncertainty. And this is kind of a very nice theory, because what this means is that through active inference, we sample the world and author our own sensations, and we can also minimize uncertainty by changing sensations in our environment, and it basically equates action and perception. And there are many depths of unexplored entropy, both inner and outer. But what psychedelics basically do is that they increase our internal entropy. And it's kind of an interesting thing to also think about how this translates to changing our behaviors, so also outwardly. But let me just give an interim summary of the first part before I go on to the actual research that we did with these theories. Basically we have now this Bayesian theorem, and then we have predictive coding that describes how this very long equation can actually be implemented across different levels of the hierarchy. And we know now that these sort of Bayesian predictions act as top-down factors that try to explain away the bottom-up uncertainty in order to sort of establish order in an otherwise chaotic system. But how is this implemented? So we're still not completely at the point where we can talk about how the brain implements these things, but we can at least talk about how they can be artificially implemented within a neural network. And what you can now do is you can train these deep convolutional neural networks to classify images. You just give them, you give the network 10,000 images, and you tell the network to tell me what the dogness of this picture is, or tell me what the houseness of this image is. And by giving it feedback, we can deconstruct its basic features of dogness and houseness. And then you can probe like the activity of what the what the receptive fields of these artificial neural networks look like, and they actually are quite resemblant of what a receptive field in the brain also looks like. So you have oriented edge detectors, and then on the higher levels you have categoric features like dogness or houseness. What you can also do is basically run this network backwards, and it produces these nice house dog images for you. So this is why they call it these, the Google Deep Dream. So an approximation of what what the network is doing when it's tripping on acid or something. But does this have anything to do with how our visual system actually works? That is kind of the question. Now what you can do is use a so-called representational similarity matrix. So an RSA, representational similarity analysis. And basically here you take the activity of the network and plot it against itself. So you just, while it's viewing classifying different images. And what you can see then is you can look at different activity clusters. So here you see the network. So here you see like the neural network looking at humans, at bodies, at faces, at animate objects or at inanimate objects, and you can compare it across the activities acquired with fMRI, where they gave humans the same stimuli and acquired brain activity from their inferior temporal cortex. Or also with from monkeys when they took activity from their from their actual neurons, and they found that the representational similarity is basically the same when you compare it from between a convolutional network and a human cortex or like a monkey's brain. So what this basically says is that these neural networks do capture, at least in some part, something that that both humans and monkeys are doing in a fairly similar manner. And what does this and how does this relate to psychedelics and hallucinations? Well, we can think of it in this way. We have a sort of sensory deprivation condition, which is the equivalent of giving our network Gaussian noise, which is our sensory information. Then we feed the neural network a lot of expectations by having the network repeatedly learn to classify ants. So it's sort of primed towards telling me what ants are. And then when I give it the random Gaussian noise, it just sort of explains away the sensory information by by processing the antness of a sensory stimulus. So you see now that the Gaussian noise can just easily become misinterpreted as ants based on these prior expectations. This also gives us some very nice processing tools. For image processing, when I take an image of yours truly and give it some expectations of a nice Alex Gray image, voila, there you have it. What you can also do is, you can stimulate different parts of these neural networks to create different types of hallucinations. So here they probe these different layers of the network, and they got distortions on different types of on different levels of the feature space. So you have here like low-level feature hallucinations, and here you have like high-level feature hallucinations. And what you can do is take a virtual reality goggles and implement images where you stimulate these sort of hallucinations and compare the phenomenology of how people experience these videos while they're seeing something like this as compared to something when they're tripping on psilocybin. And what the group of Anil Seth found was that when you compare these phenomenology, you have like a fairly similar readout of how people experience these states. I wouldn't say the two are identical, but phenomenologically speaking, they're fairly similar. So if the entropic brain theory then describes qualities of different mental states, as I said before, then this free energy principle or predictive coding describes how we arrive at these different states. So it describes the process by which we reduce then the uncertainty and sort of go between these different states. What is also relevant is this sort of rebus theory of heightened top-down processing, which recently came out from Friston and Carhart-Harris. This is sort of a similar framework that also postulates that under regular conditions you have this sort of heightened top-down processing, and they draw this analogy between imagining the brain as a sort of ball being dropped on a solid surface, and then the ball just sort of bounces back. Whereas when you give people psychedelics, this hard surface basically melts or turns into something more fluid, and when you drop a ball on it, it just creates these sort of ripples. So the hardened surface being our sort of expectations, which then become sort of softened and much more distributed, so it sort of evens out the playing field. You no longer have these very hard maximas, like these very these very strong predictions, but rather you have these sort of distributed bottom-up processes and much more fluid categories, which sort of frees up the incoming information. Yeah, 1 second. I'll come to questions in a minute. So if you take this sort of free energy account of something like a psychedelic mystic experience, so this is like a theory from Michiel van Elk, who's my supervisor. He talks about the disintegration of interoceptive versus exteroceptive awareness, so we no longer have these sort of strong boundaries, strong expectational boundaries between internal or external worlds, but rather all these categories just become one fluid continuum. And in this context, you can also view perception as a type of controlled hallucination, that the brain endogenously generates perceptual experience, dreams, hallucinations, perceptual illusions, imagination, and mental imagery, and that the same system is used to perceive the external world in doing so. Another nice way of looking at it is perception as some kind of augmented reality. So here you just have a bunch of random features, which are the moving dots, which seem fairly random until you superimpose these sort of different shapes onto them, and you can then they become something else. It's also what is also nicely depicted in this is that you can have various levels or different priors sort of augmenting the sensory information at the same time. Which varies in complexity. So yeah, again, this is the interim summary. Now, what am I going to talk about in the rest of the talk? Well, on the one hand, I think I've now drafted an outline of how you can plot out these different sort of processing hierarchies between top-down and bottom-up processing, but it's still somewhat vague and opaque what exactly top-down and bottom-up mean, like how do you exactly define this feature space. And in the rest of the talk I want to look at some experimental data about, on the one hand, how do you define these sort of lower parts of the processing category. So typically people in the field would look at faces or houses or randomly moving dots or Gabor patches. But what I'm also interested about is the notion of a hyperprior, or these so-called over-hypotheses or priors about different priors. And here the idea is that the highest levels of processing involve sort of expectations about other expectations, and these then becoming these sort of causal rules that determine how we perceive the world. So you can talk about a conjunctive prior, which is like a multi-causal model of the world, or like a unicausal model of the world. And one of the stuff I want to work on in the future is looking at how psychedelics might change people's worldview and how this could be explained based on changes in the way they make causal inferences. So, which is also related to cognitive flexibility. Yeah, that's the end of the first part. Maybe we can take some questions now, and the break later. Yeah.
Right, so one thing that isn't clear for me is which psychedelics are you talking about when you say they're implementing the effects of this? Because they have, like, on the brain, all the same reactions, chemical-wise, or, there's a difference between
Yeah, absolutely. Yeah, so generally it is thought that the 5-HT2A receptor is responsible for most of the psychedelic effects. Yeah, so the word psychedelic is not really defined that very well if you look at it from a pharmacological perspective. There, a terminology such as serotonergic hallucinogen might be more appropriate. Also because all of these substances then have affinities to other receptors as well. But usually so you can also have selective antagonists, ketanserin, where you block this one receptor and then most of the psychedelic effects are gone. And this is actually the same for most other substances that you'd call psychedelic, like DMT or psilocybin or mescaline. That doesn't mean there aren't any differences. Right, so with DMT, for example, you don't have any kind of pharmacological tolerance either. I wouldn't say that this theory accounts for all of the differences between psychedelics, but at least it accounts for what you would what you'd call psychedelic in the more general sense, though. Like when you're trying to explain what does it mean to have a psychedelic experience, for example. But yeah, there are then substance-related differences. Yeah.
What if you take the same substance and then you make more expectation about the future? Do you have more same experience about that? Because you learn things on experience, and if you have multiple experience, we see this in a pattern for worldview.
Yeah, I agree. So this is why what I was hinting at with like the active inference thing. So you can always just sort of challenge your expectations by going out in the world and feeding yourself more information. Yeah, I agree. Yeah.
One of your first slides, you have a graph that on one of the axes you have the e-body solution. I'm curious about like how is this quantified?
Yeah, so yeah, I'm not that familiar with all the sub-items of that questionnaire, but yeah, yeah, I think there are some, I mean, it's just basically a questionnaire, and then you have scores on that questionnaire, and then you can rate people on it, I don't know, and then you take the ratings of that score and correlate it to the brain activity. Yeah, I couldn't tell you what the exact items on that are, sorry. Okay.
But it's more quantitative than qualitative.
Well, I mean, it's a questionnaire, right? So you have different items, and it's from the third person perspective. Like, yeah, so it's not a first person account, right? It's a third person account.
Just a question to make me kind of process the information better. Are we referring to high doses only, or?
Yeah. So I'm not referring to anything. Well, I did do two studies where I looked at this. So one idea would be that whether on microdoses this has an effect or not. I would think that this is something that I want to look at experimentally, if like a low dose has some kind of effect, or whether you really need a higher dose, and what this could be translated into. But you could argue that maybe if there's a high dose, these effects are still there, only on a sub-perceptual level. But I don't know yet. Yeah, should we move on? Time-wise, we are quite over time. Yeah, okay. Yeah, so I can leave out the third part if we don't have enough time. Okay. Okay, so moving on to the second part. This is a little bit of a break from the other one, because then this has more to do with some of the specific details of how I conducted the research for my master thesis, which was called "The Doors of Perceived Simultaneity: The Effects of Microdosing Psilocybin on the Perception of Asynchronous Audio-Visual Speech." And this was the very first placebo-controlled within subjects designed to investigate microdosing in Amsterdam. And it was not a medical study, but it was pre-registered publicly. This raises some of the issues, the tensions between medical science and something like a crowd science, which is currently being debated amongst the psychedelic community. And recently there's been a lot of hype about microdosing, and that people think that it can translate to personal benefits and their mental well-being. But there was also this critical review that looked at it, and they point out, rightfully in my opinion, that there are not a lot of controlled studies that have been actually done so far, and there's like a serious lack of placebo controls. So, and also that the term subperceptual is not very clearly defined, and there's a sort of need for a better terminology, maybe subpsychedelic or microdosing versus midi-dosing might be a suggestion worth considering. Let's just skip through that. And anyway, let me just tell you a little bit about what we did in the study and how we approached this topic in figuring out whether microdosing is real or just a placebo. And let's look behind the veil and look at what we did. The study couldn't have been done without the help of the Psychedelic Society of the Netherlands and microdosing.nl. Who provided us with this workshop space and a smart shop in Amsterdam, where we recruited our participants and gave them instructions on how to do the microdosing. But also, the study was coordinated by APRA, which is who basically served the middleman in communicating with the Magic Truffles people who donated the truffles to us between the microdosing community and the PSN communities. And this was kind of important because this way we could convince the University of Amsterdam to let us do an actual study on the effects of psychedelics, because we served as sort of the layer between the university and being directly involved with administering psychedelics to anyone, and in doing so we just oversaw that people were doing this of their own accord, that they sort of signed up for all the workshops, the microdosing workshops independently, and that that nobody was directly implicated in administering any substances to anyone, but in a way that we could still control for the effects of microdosing in an empirical manner. Also, we had the help of the Department of Chemistry and Natural Compounds in Prague, who then later analyzed the exact amount of psychedelics being used in this study and quantified their psychedelic alkaloids. And basically, we went on to recruiting people from the general public. We had around over 300 people who were interested. At some point, we just had to make a cutoff, and because we had way too many people who were interested. Then we pre-registered the whole entire study in an open science framework. And after the online recruitment, we applied some of the pre-screening criteria. So we pre-screened the participants for medical conditions, etc. And in the end, after the pre-registered exclusion criteria, so we also excluded people who, in their self-report said that they used other drugs in this period, so not just psychedelics, but where the data could be contaminated by people who used modafinil or some kind of learning drug, for example. Then we had to exclude those participants. And in the end, we were left still with 35 subjects in the final data pool. So like 18 females and 17 males. And let's look at the workshop protocol. So basically, the people went into the workshop. They signed an informed consent form. The exclusion criteria were verified by the researchers independently. The ownership of the truffles were immediately transferred to the workshop participants. And basically the participants were then instructed on how to create these microdosing capsules. Each of them had to put 0.7 grams into 2 capsules over a period of 3 weeks. So they created 14 capsules under semi-controlled supervision conditions. Then we basically took all of the participants' self-made microdose capsules, and we randomly mixed it among all the participants so that there wouldn't be any minimal risk that the participants were using too much of it, so not actually microdosing anymore. And we distributed placebos, and we switched the 2 samples in a counterbalanced order. So neither us or the participants knew whether they would first be using a microdosing batch or a placebo batch. And then the idea was to compare their behavior from one condition to the next, to the other in a counterbalanced manner. This is what the general timeline looked like. So first we did the PSN screening, and then the university screening, and then we held the workshop where they took their first microdose, and then they came into the university to do our tests independently. So across 4 different dates, they came in and did our cognitive task battery, which I will be talking about in a minute. Then afterwards, we quantified all these psychedelic alkaloids within the truffles. So it's not just psilocybin actually that is psychedelic, but you also have psilocin, psilocybin, baceocysteine, or baceocysteine, which have all been implicated in having various types of psychedelic effects. What we did was basically we took the samples at the 4 different dates where we did the testing. You don't really see an overall consistency, which shows that you're dealing with an organic compound, which means that there's going to be a lot of variance in it. But what it does show is that the compound did not decline over the 2-month or 2.5-month period where we did the testing. So that's actually quite promising in terms of the result. But what are the aims of our research? So I wanted to look at temporal dilation, which is up to date the only actual results on the effects of microdosing or the effects of time dilation, which so people perceive small distances of time to be longer than they actually are. And I wanted to look at the effects of temporal recalibration, which I will be talking to you, explaining to you in a minute. We also looked at emotion regulation and creativity and the sense of awe. A lot of these were investigated by other researchers, so I won't be talking about that today. But I will be mainly talking about temporal dilation and temporal recalibration. So, as I said before, the effects of microdosing LSD has already been investigated by another group. And what they basically did was that they showed the participants blue circles for small random intervals, and had the participants push a space bar to repeat the same amount of time. So they had to reproduce the duration of each circle, and they found that the durations that these participants reproduced were then estimated as longer than they actually were. So that was basically our starting point. However, I would note that there are some like theoretical shortcomings that I see with measuring time perception in this manner, namely that if the perception of time is dilated under the effects of psychedelics, then how come it is not dilated when the people are reproducing it? So this is like a fundamental criticism to the method being used, right? So if they perceived it as longer, then they would also press the space bar for a shorter amount of time and just perceive that to be longer as well, right? So, how well does this actually capture what it's trying to measure? And also time as such is not being measured, but rather a stimulus, a duration is being perceived. Now, what this means is that, in a certain amount of time, we see a certain amount of things happening, and based on how fast or how much things are happening, we perceive that as more time having passed. So it isn't as such that we have like an exact mental representation of time, but rather we are registering a certain amount of changes in our environment. So we're processing the entropy, and the sensation of time passing arises out of this. And also there is no suprasecond clock in the brain, because if there was like some kind of central clock inside the brain, then all the other parts of the brain would have to be centrally connected to this in order to transfer this information across all these different sensory domains, etc. So time perception is rather context dependent and modality dependent, so it matters whether you're registering the duration of an auditory stimulus or a visual stimulus. And in relation to the entropic brain theory, it could then be said that that by increasing entropy, you're also dilating your perception of time, because suddenly there's like more amount of things that are happening in the same amount of physical time, which causes these temporal dilations under the influence of microdosing. What this means is that it changes it creates a shift in the temporal scales, where processes that are slow are then perceived as fast due to the increase in entropy. Because entropy basically spans through both time and space. Now you can imagine this as a sort of moving train, where you're sitting on a train and there are some things on the horizon that are not changing, but the things that are close to you are changing very rapidly, right? So in this sense, the entropy, or what the brain is sort of trying to reduce, can be conceptualized across both time and space. And like I said before, what this means is that within the same amount of physical time, seemingly more things are happening. So this was kind of my explanation for why I think the psychedelics in subperceptual doses might cause something like time dilation to occur. And this is what I wanted to investigate. Now, how do we investigate that? Now here's the thing, reality is perceived as a stable and synchronous continuum, even though auditory information is much slower than visual information. So light travels much faster than sound, and every time we see a moving car, it doesn't really bother us that it's further away than something that is happening right here, even though the information is taking like way longer to actually be processed, right? Also, our brain processes visual information and auditory information at very different speeds, but our brain doesn't really have a problem with that. It just constantly adapts to continuous changes across time and space, and it needs to have some kind of, basically it needs to have some kind of prior expectation about that. It has to sort of have a representation of how far away something is, and then it can just account for these sort of modality-related delays in a context-dependent manner. So I was kind of curious to see whether psychedelics change this process. It also Also, we have to update these priors like instantaneously. If there's an object that is moving, like a car, then we sort of have to update our representation of how far away that is. And a perfect example of where so a nice natural example of where that perception of synchrony breaks down is when we look at a thunderstorm, right? So there, because the auditory information travels so much slower than the visual one, we no longer perceive that necessarily as synchronous. And this means that there must be like a certain time span wherein certain objects are then perceived as occurring simultaneously. And this is what is being measured in the temporal recalibration task, which I will show you in a minute. Now, in the temporal recalibration task, what I basically did was we took a video image of a woman pronouncing a syllable, and we just created a temporal time span of stimulus onset asynchrony delays. So we just took the auditory signal and either dragged it 400 milliseconds before the visual onset or 400 milliseconds after it, and everything in between, and showed these across 700 different trials to our participants and had them rate whether they thought this was synchronous or not synchronous. This is what it basically looked like.
Ba. Ba.
Ba. Ba.
Yeah, so it's not that. Yeah, it's it's not that interesting, and you can imagine how if our participants felt after having to do that 800 times or so. But what you can do with this function is that you can plot all the amount of times they thought a certain trial was synchronous or not synchronous, and plot it across all these different temporal delays. So you can, and what you can do then is that you can change these sort of categorical variables into nice continuous variables by fitting a Gaussian function on it. This is also pretty nice because you can then perform your statistical tests based on the distribution of the Gaussians, and you can account for what the so-called point of subjective simultaneity. So you can create a psychometric function of how an individual participant perceives simultaneity as sort of a function that tells you, okay, here at the top of that curve, everybody they will say this is definitely synchronous, but and then you can look at how wide is the curve or whether this is shifted or not. Also, because the data then provides two different types of information. Namely, you can divide all the trials where the previous trial, the audition led, and all the trials where vision was leading, and you can see whether the previous information had some kind of influence on the next trial. So what is well documented is that your perception just automatically adjusts to changes in the space temporally. So whenever there's a trial where audition was leading, normally what would happen is that this Gaussian curve would then sort of shift. So in all the trials where there was a larger temporal lag before, on the next trial you would see like a larger shift in that Gaussian function. So the basic idea was that if psychedelics then increase the entropy or increase the temporal resolution of the stimuli, people would then also perceive these sort of temporal lags as lasting longer in their duration, and that is why I did the experiment. Now going on to the results, or rather the lack of results, we did not. Unfortunately, we did not replicate the temporal reproduction task, the one I was talking about with the blue circles. Also, what we found was that there was no shift in the point of subjective simultaneity, which is looking at the mean of the Gaussian. We did find that under psychedelics, people were, or under microdosing, people would still adapt to it. So so what we see is that here are all the trials where audition was leading and all the trials where vision was leading. And they sort of shift in a context-dependent manner. However, this is just basically the same across microdosing and placebo conditions. What does change under the effects of microdosing is that the overall shape of the Gaussian becomes much wider. So there's like an increased bandwidth of the perceived simultaneity, which is kind of speaking in favor of having time dilation under the effects of psychedelics. So there's basically a wider range of perceptual events that a person will be willing to accept as occurring simultaneously. However, it does not really affect how people put this into context. So the results are still kind of ambiguous on that. On mystical experience, this was another hypothesis being tested by my supervisor, Michiel van Elk. Basically, the idea here being that people under microdosing would have a higher probability of experiencing these moments of awe and oneness with their environment. You could say that, well, if it is subperceptual, then why would people have these experiences? Well, according to this other study that he conducted, he also showed that you can induce these sort of effects also by just showing people awe-inducing videos. So that is what we did. I can show you one of the samples of the videos that we used in the experiment to lighten the mood a bit. Everybody's feeling in awe now? I hope. Well, some people like it. Others found this task a little bit annoying. But anyway, we didn't only just look at awe-inducing videos. We also wanted to compare it to positive or funny videos. You want to see an example of that as well? Okay. Good stuff. Okay, and finally we had our control condition where we looked at just neutral videos. Let's just look at that as well. Yeah, some mystical tractors. Yeah. So, measuring the sense of awe, you asked about how we measured the ego. Well, here, luckily, I have some of the items. So here are some of the questionnaire items that we use to measure the sense of awe. Like, to what extent did watching the video influence the experience of something beautiful, or the feeling that ultimately all life is one, or induce feelings of self-transcendence, or to what extent did you experience a loss of sense of space and time during watching the video? Were you impressed by watching the video? What did we find? Well, we found that under the psilocybin condition, you had an increase of the sense of awe that you can also see across the awe condition, the positive video condition, and the neutral video condition. So it was like strongest in the awe condition. So they scored significantly higher under sense of awe. However, you know, was this a mystical experience or a social or expectation at play? We also looked at sort of the blinding accuracy of the microdosing trial, and found that participants were much better at guessing their condition in the second session. So, you know, they became much more aware of the fact that they were microdosing, which is, in that sense, you sort of have to look at these third-person accounts of people's experience with a pinch of salt. So, if, for example, in this case, you could argue that the awareness of knowing that, okay, now I'm microdosing, so I'm expected to give these ratings is maybe much higher, which is, you know, might also be a reason why people scored in this manner on the questionnaire. But, you know, anyway, interim summary, we managed to organize an academic study on microdosing independent of any external funding. So we paid exactly €0 for it. Microdosing does not affect temporal adaptation, but it does dilate the bandwidth of perceived simultaneity. You know, evidence for time dilation, but we also have evidence against time dilation since we didn't reproduce the other study. Placebo condition remains crucial as our participants improve their guesses accuracy from 60 to 70% after switching their treatment condition. Yeah, so I guess I had a third part, but I already